and thanks for the invitation to connect to this audience. I'm going to tell you about some of our latest work on autism and the human microbiome. I have some disclosures for commercial interests. None of them are particularly relevant to this talk, but I have to tell you about them. I'd like to start by inviting you to consider what you saw when you looked in the mirror this morning as you were getting up and getting ready to start your day. For myself, I saw an organism that's just 43% human, and not just because it was early and I hadn't had my coffee yet, but when we think of what makes up our body at the level of cells, each of us has about 30 trillion human cells, but we have about 39 trillion microbial cells, mostly bacteria in the gut. So by that measure, you could say we're just 43% human. Now, you might think, well, it's the 21st century. Shouldn't we be thinking about our DNA instead? So let's think about that for a moment. To our 20,000 human genes, astonishingly, the size of the gene catalog in our microbiome is 2 to 20 million microbial genes. And so when we're thinking about this at the genetic level, you could say that we're at best 1% human. And so what this means is that despite all of the effort focusing on the human genome, we're ignoring 99% of our genes when we do that, when we don't consider our microbial component. But what's more astonishing to me is that the 99% we ignore are the 99% that we can change. And taking control over this process of changing our microbial genes and their functions is something that could have tremendous implications, not just for autism, but for health generally. So through advances in machine learning over the last 10 years or so, we can now build predictive models for many traits related to health from the microbiome. And for example, I've been working on obesity for the last 15 years. And today I can tell you with about 90% accuracy if you're lean or obese, based solely on the genes of the microbes in your gut. So on the one hand, this is a cool trick from a technical perspective. On the other hand, we don't think it's going to have a lot of clinical significance because I bet you can tell which of these people is obese without doing any sequencing at all, right? But on the other hand, if we try that prediction task, lean or obese, from human genes, we can only do it with about 57% accuracy, whereas we can do it with 90% accuracy from microbial ones. And we're learning that the gut microbiome is an incredibly sensitive data recorder, including information about how we're born, like our delivery mode, how we're fed both early in life and later on, diseases we have, drugs we take, even exercise we do, and the locations where we do that exercise and where we live. But I'm mostly talking about the gut microbiome here. It's important to remember that the gut is not Vegas, and that what happens in the gut does not stay in the gut. And the gut microbiome, we're learning, affects processes throughout the body, including digestion, the immune system, protection and clearance of pathogens, and brain development and behavior. So those are all positive effects of the gut microbiome. It can also have negative effects, though, including metabolic syndrome, gastrointestinal disorders, cancer, and most relevant to this talk, neurological and neurodevelopmental conditions. So the microbiota affect neurodevelopment through the gut-brain axis. And of particular, there's a lot of different ways that it does this, including neurotransmitters, neuropeptides, microbial byproducts and metabolites, cytokines, and bacterial debris that can stimulate the immune system in different ways. And Sarkis Mazmanian, my good friend and a collaborator, has been for a while looking at this model of maternal immune activation in mice, where essentially what you do is you inject mice with double-stranded RNA that simulates a viral attack, and the offspring of those mice have a whole lot of behavioral abnormalities, including impaired social interaction, decreased communication, and so on, and also a series of neuropathologies that resemble human autism in some respects. So Sarkis published a groundbreaking paper in Cell in 2014, where what he was able to show was that the microbiome was causing a lot of these effects in mice subjected to the maternal immune activation model. And so what it was able to show is that treating the mice with a naturally occurring microbe, a probiotic, if you will, from the human microbiome, was able to alter the composition of the gut microbiome, improve the barrier of the gut lining, reduce the leakage of particular metabolites, especially one called 4-ethylphenyl sulfate, or 4-EPS, and then ameliorate a bunch of the behavioral abnormalities seen in this model. And so in a commentary on this paper, we proposed that this could be a very general strategy towards coming up with better probiotics and other strategies, not just for autism, but for other neurodevelopmental disorders, where you start with this kind of mouse model work where you see a difference between healthy and sick, You do preclinical trials in mice to look at the effects of antibiotics, prebiotics, probiotics, to reconfigure the microbiome into a healthy state. And then ultimately start to develop a human analog of that preclinical treatment. So one of the standard rating scales for autism, as you know, is the Childhood Autism Rating Scale, or CARS. And this is used to assess the severity of autism symptoms in children, looking at communication difficulties, motor difficulties, social impairments, repetitive behavior, and sensitivity to change and stimulation. And one thing that was completely remarkable to me is work done by a consortium of labs in Arizona, where what they did is they took 18 children with ASD and they gave them a 10-week fecal transplant treatment, and then continued to track their autism symptoms using CARS over 2 years, where those autism symptoms were significantly improved 2 years after the fecal transplants were done. And I just want to say a little bit about fecal transplants. So this is a map of the human microbiome that we put together as part of the Human Microbiome Project, where the different parts of the body, like the mouth, the skin, the vaginal, and the fecal communities, emerge like different continents on this map. And fecal transplant is mainly used for a disease called C. diff. And patients with C. diff have severe diarrhea, and you can see that their microbial communities, these orange spikes, look nothing like a healthy stool community, even though we're looking at stool here. And what's going to happen is 4 of these patients are going to get a fecal transplant from one donor. And to give you an idea of what fecal transplant is, this is Bill Sanborn, who used to be our division chief of GI here at UC San Diego, about to deliver his first one with a huge grin on his face and a preparation of hospital-grade stool from a nonprofit called OpenBiome, because the FDA does regulate stool as a drug, so you shouldn't try this at home. But anyway, 4 of these patients are going to get a fecal transplant from that one donor, who you could see was in the healthy region in brown. And this is work we did with Alex Kreutz and Mike Sadowski at the University of Minnesota. And what I'm going to do is I'm going to animate the microbiome journey of those 4 people through time. And each frame in this animation is one day in the lives of these people who had had severe diarrhea for years to be eligible to participate in this trial. And what you can see is that immediately, in just 2 or 3 days, their entire microbiome ecology resets from the sick state into the healthy state. You can see that they stay in that healthy state through years of follow-up. And coupled to this, all of their diarrheal symptoms are gone. And in general, fecal transplant for C. diff has been 90 to 95% effective in clinical trials, whereas antibiotics are only about 30% effective for recurrent C. diff. So this is a real game changer for this life-threatening disease. And so what we're trying to figure out at the moment is, for other diseases, how can you identify a problem with the microbiome and then guide it back into health? And one thing that's especially exciting to me is that this works for autism. So the first paper on this in 2017 is this open label study I was talking about of 18 patients with autism. And what you can see is that their GI symptoms on the left, as well as their autism symptoms like CARS on the right, were significantly improved both at treatment and 8 weeks after, and 2 years after. And so this seems to have a durable effect on improving autism symptoms. And so the question now is, for what other diseases can we also do this for? Because there's a huge number of different diseases that have been linked to the microbiome, where essentially what we need to do is find the good places and the bad places on the map, and then go beyond that to develop a kind of microbiome GPS that tells you what you can do to get from one location to another, whether we're talking about something as extreme as fecal transplant or phage therapy, or something as gentle as diet, which over the long term we've shown has a massive impact on reshaping your microbiome. So I want to say a little bit about that. So we've been turning to citizen science to understand the microbiome through the American Gut Project, where we've sequenced over 30,000 samples provided and funded by members of the public, although we still need more. And this has developed into the Microsetta initiative. For the last few years during the pandemic, we've largely been focused on getting kits out to large cohorts to study the effect of SARS-CoV-2 and long COVID. But we're getting back to opening it up to the general public right now. And one thing we can do, having tens of thousands of people with exactly the same questionnaires and the same microbiome assays, so we can start to figure out what has a big versus a small impact on the microbiome. When we look at the number of people per group you need to find a particular effect, The steeper the curve on this graph, the bigger the effect is. So you can see that things like age in brown, whether or not you have inflammatory bowel disease in red, and antibiotic use in purple are all strong effects where you need a few dozen people per group to see the difference between those. And then things like how much you sleep, how fat you are, how much you drink, how much you exercise have more subtle effects, where with hundreds of people per group, you can see the differences between them. But what astonished us was that the number of types of plants, just the number of different species of plants you eat in a 2-week period, has the biggest effect that we saw in this preliminary analysis on the gut microbiome, if anything. And that's something that's totally under your control. So the American Gut Project is a platform where we've extended it internationally. We've worked with patients in ICUs, in depression, in students, with museums, and early on looking at autism patients. And one particularly productive collaboration we had was with the Pitt-Hopkins Syndrome Society, where these are individuals who have a mutation in a human gene, TCF4, and they often have moderate to severe intellectual disability with distinctive facial characteristics, and where epilepsy and GI problems are common. So we collected samples from Pitt-Hopkins patients and family members to explore genetic links on the microbiome specific to Pitt-Hopkins or different with unaffected family members. And what we found was that a particular organism called Clostridium baltiae was especially enriched in individuals with Pitt-Hopkins syndrome. We also saw that this seemed to be age-mediated. So this difference that we saw was also strongly correlated with age, where the older you get, at least if you're healthy, the less C. baltiae you have. But that doesn't seem to be true if you have Pitt-Hopkins. So this is an example of the way that you can find associations between particular microbes and particular forms of autism. I should mention that we also found some mechanistic data with Peter Dorstein's group, where there are particular bile acid modifications that seem to be enriched in Pitt-Hopkins. And so bile acids have a really important role in regulating the immune system and bacteria generally. And this is a very exciting emerging area that we would love to apply to other aspects of autism, but haven't done yet. But so this is a great example of how you can look at an individual form of autism and look at the particular bacteria that are involved in that. But there's a problem, and this is familiar to many of you if you've had microbiome profiles done, especially if you've had them done by different labs, is that even if you have the same sample, even if you have the same data, if you use different software to analyze it, you can get completely different results. So each row across this diagram is one person from a cohort that we looked at to look at microbial associations with disease. We took exactly the same data and ran it through a whole bunch of different algorithms. And these are classified at the phylum level, so very broad groupings of bacteria. And you can see for the same row, depending on what software you use, you can get a completely different picture of what's in that sample. And they're not even correlated with each other across programs sometimes. So, especially if you're a physician, what are you supposed to do with this kind of inconsistency? So Jamie Morton, one of my former graduate students, who's now at Guts Inc., a startup that he's founded to do this kind of thing, led a remarkable paper in Nature Neuroscience that came out this year that reconciles this problem of different analysis methods giving you different results. And so what he found is essentially doing very carefully age and sex matched differential abundance analysis using log ratio analysis allows us to integrate a lot of different kinds of information from different studies for the first time. And so this multi-level analysis essentially compared children with ASD to neurotypical children using differential analysis, looking at the abundance of microbes in the gut. And then this was repeated for a whole lot of other kinds of data, like the metabolites that I showed you before, and then compared the results across different studies. And so what we find there is that we find that there are autism-specific metabolic pathways that are reliably associated with the gut microbiome, and these are reproducible across different studies if you analyze those studies in the right way. with 16S ribosomal RNA profiling and shotgun metagenomics integrated for the first time with our Green Genes 2 tool. And the heat map just shows the comparison of the abundance of microbes between the two groups, where many of these associations are now reproducible, whereas they weren't before. So this study reveals significant microbe differences in ASD versus neurotypical kids. And I won't go too much into the details of this, but essentially the ASD kids have far more inflammation-related microbes. One of the exciting things about this is that there's a great potential to treat this with anti-inflammatory diet, as we're already finding in adults in other studies on endometriosis, on inflammatory bowel disease, and on rheumatoid arthritis, and most recently on lupus, where for all of those conditions, we see significant improvements with anti-inflammatory diet. And given the inflammation-related microbes that we're seeing reliably coming up with ASD, this may be a useful dietary intervention to use in ASD as well. So with that, I'd just like to say thanks again for listening. Thanks again for having me here, and really looking forward to the Q&A. You can find out more about this kind of thing in my books, Follow Your Gut and Dirt is Good. Thank you.